To wrap up the virus segment, let's go over the question of, are viruses alive? There are arguments against them being alive, and there are arguments for them being alive. Let's go over against first. Viruses carry on the process of life only inside of cells. Therefore, by definition, they are not really alive. They have to have a cell to be able to reproduce, make more of themselves, and they don't have an independent metabolism. They're not cells. Part of the definition of life is that life is composed of either a cell or cells, and if you're not a cell, you're excluded. Now for the arguments for them being alive. They have sophisticated methods of invading cells. In fact, depending upon the virus, they can tell if a cell has already been invaded by another virus, and they can detach from the cell and go on and look for, so to speak, another uninfected cell. So there are control measures happening outside of the cell. They take control of the host cell. They take over the metabolism of the cell. They have genetics, so on. And with some viruses, including hepatitis B, metabolism happens outside of the cell. The genome replication is finished inside of the capsid in the virion. So you can tell which side I'm on, but there are arguments that they are not alive, that they are infectious chemicals, or you can say that they're the ultimate parasites. Now, on to virioids. Virioids are small circles of RNA that are infectious in plants. Now, you're looking at these virioids and you're going, that doesn't look like a circle to me. Well, they've done some hydrogen bonding between the different sides of the circle, so they kind of collapse down. Here is the genome of a phage to give you a size comparison between what a virus genome looks like and what a virioid genome looks like. Virioids are considerably smaller, and they cause several plant diseases in crops. These are some potatoes that have been infected with the PSTV virioid, and as you can see, if I were a farmer, I would be very unhappy getting this rather than this, which is a sellable product. One would think the virioids were as small as you could go as far as infectious particles, but now we're going to talk about prions, infectious proteins. In mammalian cell membranes, we have what is called PRP, and there's cellular PRP and prion PRP. Now, we don't know exactly what PRP does, but it is vital to the functioning of the brain. If it's not there doing its job, neurons die, and you end up with holes in the brain. Now, the properly functioning cellular PRP has these alpha helices, whereas the prion PRP has these beta-pleated sheets. Now, this structure is extremely stable. It can't be denatured by heat. It can't be broken down by acids. The only way to get rid of these is through complete incineration. Well, let's talk about how we get prion PRP versus cellular PRP. There are two ways of doing it. One, you can have a mutation that causes the prion PRP to be present and coded for instead of the cellular PRP. Slowly over time, the prion PRP builds up, it's non-functional, the neurons are not able to live, and you start getting enough of these holes in the brain that the mammal can no longer effectively function, and it develops neurological problems and it dies. We used to call these diseases slow viruses because we didn't think there was anything beyond viruses, as far as being smaller, that could cause disease. And there are a couple of these diseases. Scrapie in sheep, it's called that because when they start getting the neurological problems, they will find a tree or a fence post and they'll rub up against it until they scrape off their wool. There's also chronic wasting disease in deer and elk. That's found in this area. Then there's Crutchfield-Jakob disease in humans. All of these have been linked to genetics. Well, we started finding in about the 90s, young patients that were showing a variation on Crutchfield-Jacob disease. And about the same time, we started having neurological pathology showing up in cows, and we called that mad cow disease. Its official name is bovine spongiform encephalitis. And that's when we found out that prions could be transferred from the organism that has the genes for coding for the prion PRP, and it can travel through the digestive tract, through the blood supply to the brain, where it can start converting cellular PRP into prion PRP. Prion PRP enzymatically, if you want to think of it that way, forces cellular PRP into the prion shape. Eventually, they figured out what was going on. If you feed animal protein to herbivores, they gain weight faster. Makes sense. Animal protein is easier to digest. It's just herbivores don't chase down other herbivores to eat them. So what they would do is they would take the parts of sheep, goats, cattle that people don't want to eat. Brain tissue, nervous tissue, hooves, nose, you name it. Grind it up and put it into animal feed. Well, when you put neurological tissue from a sheep that has scrapie into the food of a cow, eventually the cow will develop mad cow disease because the scrapie prion has gotten in and started causing these holes in the brain of the cattle. When people eat infected or eat cows that have bovine spongiform encephalitis, we pick up these prions, which are not broken down by your stomach acids, and they're able to cross the blood-brain barrier, and they would develop this variant Crutchfield-Jacob disease. Because of this, we keep an eye on animals that have neurological problems. We don't use what are called downer cattle in human food. Just to be safe, all beef is no longer accepted for export from Great Britain. And we monitor herds here in the United States. There have been 2 cases of positive identification of mad cow disease in herds in the United States, but the affected animals never made it into the human food supply. But it's something to keep you up at night anyway. That's it for these topics, and that's it for this chapter. And here are reminders of what the topic objectives for these series of topics are.